problem. Yeah, a lot of potential for soil. Oh, there's a couple of hands up.
Oh, okay. Hello.
You thought you could
Sorry,
offer.
has someone done paleoseismic analysis on skeleton?
Paleoseismic. Fantastic. Sounds great. I don't know. Maybe Beth. Did you know about that?
Never heard of anyone who's not seen that. Have you heard of it? Seen other countries that have done that?
Where does the bromine come from in the knowledge? Is that an aerosol-based thing, or is that from the weather of glass, or from acids coating the glass?
Very good question. There's where's the bromine coming from normally? It's from marine aerosols, very much. That much bromine coming in one big chunk and then going away again? I don't know. There's a guy called John Dredge who's doing aerosols, studies of aerosols in caves. And rather than regrettably for him, he's basically found that aerosols don't have any influence on the trace element geochemistry. So I would suggest that it is a genuine drip water signal. But the source, probably need some, it's pretty hard to say, isn't it?
For the weather glass,
Yeah.
that was the same year as the eruption, or is that the chronology good enough to tell them?
Chronology looks fantastic.
So it's got to weather and precipitate in 10 minutes.
Well, I think the time slice that we're looking at there wasn't quite 10 minutes. Yeah.
Anyway,
I didn't publish the paper, I'm just saying.
No, it's not. Really, because I want to do it. I want to do it.
Yeah, good.
I want a trip to Melbourne.
Okay. Why don't you take me? You get someone to do my teaching for me.
Okay.
Ask the supervisor.
So, last but not least, don't you dare labour me with anything. Troy Baisden, which is going to talk about some things you can get out of soil. The thing that you're first standing on, the top of the geological. Okay.
And indeed it is. Potentially, paleopedology is just a little bit obscure. But really, it's all that we have left to talk about. It's everything that remains, I suppose, for us. And just how obscure is it? Well, it actually turns out that Vladimir Nabokov, in this sort of famous book called Lolita that you might have heard of, actually used it as an example of just how obscure something can be. You know, it's right up there with alien harps, whatever the heck that is. Wow. But really, so really, across a place like New Zealand, wherever we have this landscape with sheep or cows or things like this, and hopefully a few rainbows to go with them, we have a landscape that fundamentally the soils actually preserve a lot of the soil-forming factors, and they're an integrator of all the processes that you might care about as quaternary scientists, especially when you're probably, at least at some point in your career, trying to get a job working on contemporary systems as well. I'm going to give you a really quick half-hour run-through of everything from the really important sort of like last 100 years of pedology as it applies to paleoenvironment, right through to the hot topics of today. So to get back to the really simple textbook Soils 101 stuff, soils are an open system, and they really represent, we can summarize almost everything that happens as a set of additions, removals, translocations, and transformations. That is the essence of a conceptual model representing what happens with soils. We can also think, based on the Hans Jenny model of state factor theory, think about the soils as an independent system on the landscape that where the entire environment that provides us with soil development determines what's there, how it responds to change, and we represent that as these factors. The properties of the soil system are defined by climate, organisms, relief, parent material, time, and potentially other things. But in almost all cases, we can represent this sort of material. What I've done also, this highlights an enormous amount of literature, and what I've really provided you with for this lecture is essentially a reference list of pretty much everything I'm talking about if you want to get deeper into it. What can you actually do with that? So climate, relief, if we just want to think about what climate means, if we see a soil that actually represented a past environment, how can we interpret that? Well, it turns out that a soil, if it's in a humid environment where potential evapotranspiration, the red line, E, is hardly ever greater than the precipitation, well, that's a humid environment. And we can go across to semi-arid environments and arid environments where the evaporative signal rarely, if ever, exceeds the precipitation signal. And what happens then? Well, things that form in soil, like clay horizons, like carbonate horizons, will be deeper in this soil, if they form at all. Carbonate will be washed right out the bottom. Clay will be washed deeper than it will in this soil or that soil. And so, immediately, if you see a soil profile that represents the past, there are certain things that you can begin to infer based on what you see. You can also use slightly fancier gear. You can look at the distribution of elements. So as soils weather, they will leave behind iron, titanium, elements like that represent the initial composition. And elements like calcium and sodium will leach first. Elements like silicon, magnesium will leach quickly following them. And the rate at which those occur reflect climate. They reflect the amount of water, the temperature, and so on and so forth. And we can also see the same things as we move from temperate to tropical. If everything else were the same, we'd see a buildup of iron oxides as we move to more tropical soils. Same thing with organic matter. As we build up to a certain degree, we get to more organic matter versus sort of buff-colored soils that represent young environments. So there's a lot that you can actually see and interpret just from being out there, from looking at a soil profile from the standpoint of a pedologist. And you can see it not only at the profile level, but you can, if you auger or walk around or look at a map, you can see it at the landscape level. This is almost a very charismatic landscape. It's actually the outwash deposits of a very charismatic landscape. This is what's immediately downstream of Yosemite National Park. So you can think of this as the sediment that allowed El Capitan and Half Dome and those beautiful mountains to exist by creating a valley underneath them. And as we go through glacial episodes and back into the sort of pre-glacial environment, 1 million years ago, we see a sequence of platforms in the landscape, terraces or fans that have built up, and the soils, and these can actually be mapped as a basis of the soil properties across that landscape. And in fact, this is the type locality for doing that to a large degree. Within any soil that's old, and especially ones that are preserved, you may be able to see things that represent the plants that once lived there. So different types of plants will have different sort of root systems. Trees will be different from grasslands. And what's particularly interesting for places like New Zealand and the tropics is that palms and things like that have pretty distinct rooting systems. You can have adventitious rooting systems like you might see on a Putakawa. So you can see things about what kind of vegetation lived in the past if you're just able to follow those root channels through the soil. Now, here's a quiz. This is a paleo soil sequence. What do you think happened here? So this is actually on Easter Island, where we might be very interested in paleosols because it's the only piece of land that preserves the environment for thousands of kilometers around it in a really unique part of the Earth's climate system. And this is actually a nice datable thing. This is a basalt flow that cooked the soil underneath it that it came across, so that's why it's red. And then we have more recent soil piled up on the top of it. We can actually access that and sample down across it, probably across hundreds of thousands of years of climate history. And within each of those layers, so here you can see the baked layer in the basalt, you can see palm roots from the now extinct palm tree that was eliminated by the first Polynesians that lived on Easter Island. Over the first few hundred years they lived there, this large wine palm went extinct, but you can see these palm roots, and they follow root channels that are very different from other kinds of tree roots because they're the same width all the way through the length of the root. So you can see I've started using USB items for scale. I'm trying to keep it as modern in my research as possible. We can also see in a place like that, in terms of Chris Moy's talk, there's a few lakes there. That's one of the really great things about the island is there's actually several different areas at different elevations and environments to do the kinds of things that you learned about. And on the shore of a lake, and actually just above it, you see these things, which are kind of fascinating. And I realized that my colleagues had always thought these are volcanic features, and I said, no, those are fossilized rhizomes. So these are the fossil ones, and these are what's growing there today. You'll notice the modern ones appear to look a little bit healthier. But these are, and these probably, lived when it was cooler. And, these are from these tall grasses that grow on the edges of the wetlands on Easter Island. And they preserve these sort of rooting systems, where you've got a rankoma probably in the center that transmits oxygen. We also had fern rhizomes preserved in the same deposits. Whoops, we'll just hang on to that for a second. And so what you can learn about this is just the elevation that the lakeshore was stable for a long time. And that in itself potentially means a lot, as Chris Moy talked about, for paleo environment, but it's recognized by the paleosol that formed around that lake. Right. This emu would like to ask you a question. I've discovered in past years that people both need encouragement and a hint. This type of material, if in fact it was a real egg and not chocolate, could help you understand the answer. It's a little bit difficult, I'll admit, but if you imagine that emu, or a bird like it, some ray type, say, living in Pakistan, and you want to date the uplift of the Himalayas, an egg forms at body temperature. Soil minerals form from natural water in the environment at environmental temperature. And so knowing something that formed at a known temperature, the temperature of a bird like that today pretty much, allows you to actually work between the isotopes in clay minerals and the isotopes in the eggshell to be able to back out both the temperature at which the clay minerals formed and the water isotopes in both the eggshell and the clay minerals. And being able to measure a change in water isotopes as a mountain range is uplifted in the way of the rainfall that gets to the emu allows us to identify when that uplift actually started happening in geologic history. I'm sorry I didn't give you a good enough chance to win an egg. Any takers, or should I eat it myself? I'll just save it for next year. Okay. There's some other really great things we can do, and moving more into the contemporary, we've got to understand this global carbon cycle thing. We must do something about it and stop global warming. And you might want to try to compare the global carbon cycle as it stands there to New Zealand. One of the key points, really, is that in either one of them, there is an awful lot of carbon in soils. It's a big reservoir. It turns over slowly, so slowly that you want to use the long-term types of experiments that we talk about rather than the short-term types of experiments that agricultural or ecological scientists usually use. And so this is for New Zealand. We can see the same applies to New Zealand. We actually store an awful lot of carbon in our soils compared to, say, our vegetation, yet we're able to get carbon credits for this. Perhaps we get carbon credits or something for our soils if we better understood them. A particularly interesting problem is that relative to the size of some of our fluxes, we actually lose a lot to sediments and potentially the atmosphere. So there's some big fluxes going on there that we'll talk about more in a minute. I'm just going to jump through that one and say, oh, you're not even timing me. This is horrendous. Who knows? Who knows what'll happen? Yeah. So this is perhaps where isotopes comes in. Is we often want to think of things simply, and the amount of carbon we store in a system in the past or the present is the balance of inputs and outputs. And we can do different things. We can track this system with carbon isotopes, for example, radiocarbon, and that helps us understand how long stuff stays in here. We can also look at things like delta N15 and look at what's happening with nitrogen in the contemporary or the past environment. And whenever we do that, we might want to write equations that represent each of these processes as a term. And that's essentially a lot of the research that I do. So an example of that was actually set up by the person that founded this facility. He recognized, along with Soil Bureau, that you could go and look at soils that formed essentially in the same climate, like these two soil series here. So we've got the Egmont soil, that's by the big dairy factory in Hawera, and Tokomaru is the soil just out of Palmerston North. And if we look at those two soils and look at how much radiocarbon they took up, I did that thing with the math I talked about, and I drew some lines through it. But you can see that either way you draw it, the andesol, the soil in the volcanic terrain, took up a lot less radiocarbon from the bomb spike. You learned about the bomb spike already, right? Good. Took up a lot less radiocarbon from the bomb spike. And what that means is that this soil has a residence time that's actually much longer than this soil. So the carbon is staying much longer in this soil than it is in this soil. That's a difference in two soils based on what they're formed from. We can also look at something that matters quite a bit today. We could go to a long-term experiment like down in Winchmore. They've irrigated the same plots the same way since about the late 1950s. And we can look at plots that receive different amounts of irrigation, and we can see that, sure enough, you irrigate stuff, it grows more grass. New Zealanders like that, right? I think sheep like it too. But it actually changes the residence time, but the big change in residence time is just being wet at all, not so much here. And so what's interesting is we can start to try to understand why it is that this site stores considerably less carbon than this one at 10% irrigation, yet grows nearly as much grass. So that's useful. Another thing that we might want to do in soils, especially if we wanted to actually publish in Science, as it turns out, is to go climb up in the Southern Alps and use that beryllium-10 stuff that Kevin Norton was talking about. Really great soils problem. And so basically, if you want to try to understand the rate at which soils form, this is a great way to do it. And what they were able to do in this paper, which just came out this year, was identify some of the highest rates of soil formation in the world. And they argued that that actually helps inform our understanding of the global carbon cycle. And it's yelled a bit here. And so that, in a sense, is a really nice model for things that, while soils might seem obscure, as I mentioned at the beginning of this talk, and kind of complex, if you can actually figure out how to do something really simple with them, it basically says, we measured this, and then we drew a graph that showed that we could extrapolate it across the entire world, then you can get your paper in Science and say that it matters for the global carbon cycle. Cool. There are some other issues, though, and it means that you can still actually make a contribution to this field. I'll just show you what they did, which they climbed up a bunch of ridges like this, or actually maybe they helicoptered, I don't know. I have a suspicion they might have. But, and then they measured some points and calculated based on what Kevin told you about. And you could ask him about more if you want to work on this type of stuff, or you could work with Peter Almond at Lincoln, who did this particular work. You know, you get these really high rates of soil formation. What's interesting, though, is that if you think about it in the context that, well, I just showed you those sites, right? If you wanted to try to get the highest rates of soil formation anywhere in this landscape, those would be pretty much the places to do it, right? You know, perfect, nice, wet place, right on the ridge top. But if you wanted to look at what the entire landscape is doing, you might look at rivers draining that landscape, and in fact, different papers have done a bit of that. I was involved in this one where we looked all around the country, and there was some other work that I was able to draw on that looked specifically at the kind of rivers draining that landscape. What's interesting is that what they said about the global carbon cycle is only part of the global carbon cycle. They talked about weathering. So weathering consumes carbon dioxide out of the atmosphere and sort of puts it off in the ocean, and a lot of the CO2 that was consumed gets buried in the ocean. But there's the second thing that is kind of uniquely buried in the ocean in landscapes like this, and that is the organic carbon, the black and brown stuff in our soils. That ends up in these rivers when they look like this, and when the river looks like that, in contrast to flat places that most people can imagine, where the stuff that goes down a river that doesn't look like this usually ends up back in the atmosphere. When this happens, most of that stuff ends up buried thickly in the ocean and stays there. And so it also represents a big global carbon sink that looks like that on a map. We also have areas where we've enhanced the sink due to land use change that look like this and this on a map. But what's really interesting is if you look at the part of the global carbon cycle that science paper said was really important, well, it actually turns out that our calculations of particulate organic transport and burial are much bigger. It's just spectacular. So the two overlap down here, where we're sitting today, about a meter and a half of rainfall. But we're talking about something completely different if you can try to imagine how soils and streams and burial processes function in a landscape like the Southern Alps. So I'd like to sort of draw this to a close. We're getting there in terms of talking about everything that remains. And the point I'd really like to make to you is that rather than thinking of soils as being rather obscure, they're actually kind of a nexus where a lot of really important things come together. And if you can get something that really represents what goes on in that nexus, then you can get something really important that helps you understand past environments. The main thing I'll talk about that remains is biomarkers. Biomarkers are things like this. They're compounds that represent life. And so these are these, do you want to say that? Glycerol dialkylglycerol tetraether lipids. These things are the cell membranes that actually keep archaea, bacteria and archaea, organisms that live in soil, kind of keep their walls together. And they actually allow things to be transmitted back and forth through. These things are weird, but they have slightly different structures that turn out they vary with some different things, one of which is temperature. So somebody can go out, measure a lot of them all over the place, and calculate an estimate of how they vary with temperature. Great, and then you can reconstruct, and so on and so forth. There are some better proxies. That's one of the newest, coolest, trickiest ones that you can play with. But the key thing is just to point out that these things happen, and to really interpret them properly, we've got to understand things like when are these organisms in soil versus in the lake that you're measuring? How did they wash in? What time of year did they actually live? Or what time of year were they active? Because that's what they're going to represent. Great thing about that particular compound class is they're relatively abundant, so we can potentially measure radiocarbon, stable isotopes, and the same compounds that you're using as biomarkers for temperature. So you can get the whole of the environmental signal out of one set of compounds. That's neat when it works. And in terms of how we do that, we've been also making progress, and just so you can imagine, we can potentially relate the isotope ratio of rainfall back to daily or monthly climate data now. So this gives you a sense of some recent work that we've done where the sites were all set up by Otago University, and I've provided the modeling that allows us to calculate this kind of map where we can now predict. You can see that the colored points are more or less the same as the background map behind, where the background map represents our long-term average calculations, and the points represent the actual data. So it's not perfect at a monthly time scale, but over longer time scales it's really good, and even at the sort of time scale of months, we could do things that probably somebody that cares about the economy is really interested in. So even though you might, as a paleoclimate type of scientist, want to get back to paleoclimate, we've been able to relate climate, which is to say the rainfall across New Zealand at these points, directly to the delta D, the hydrogen isotope composition of milk that's exported from New Zealand, which helps us identify, we think, milk that turns up, say, in China that's not ours versus ours. And so if it is ours, we can figure out what part of New Zealand it came from. So there you go. I think those are some of the things that we can do with soils. You can decide for yourselves whether they're a missing nexus. But the two things, a lot of fancy tools are great, but they usually miss the point if we don't have that interpretation of the landscape and the soils that help us make sense of it.
Is there any questions before you embed?
Still at 5 minutes.
Oh yeah? Jeez, I should have left those extra slides in.
It's very interesting, that study on the soil formation rate, but it's all in vain, as you showed in the next slide.
Yeah.
And that's the last place I'd go to measure soil formation rate, because it's all in vain. Be gone next week when they have the next storm.
Yeah, but then you can form another 2 to 10 millimeters after that.
How thick are the soils? What are they? What's the?
There's pictures in the
bang
supplement.
for your buck?
You should look at the pictures in the supplementary material.
Great.
You can see the roots growing right into the bedrock, and that's actually the key to the process, in a sense.
But Pete, you're right, just to emphasise, he's quite right, soils are the most complex ecosystem on the planet. And they should be looked at.
Any other questions?
Sorry.
Any other comments?
While you're thinking of them, I'll just throw in one more thing. I brought along a banana to remind myself to say something, since I apparently have more time. And can you just imagine if I took this banana and let it decompose on soil? Do you think there might be anything left behind in the soil? This sort of went with my biomarker comment.
Yes.
Probably.
Not for long.
Interestingly, the starch grains in a banana can stay behind. And it's really just that point alone should change your insight about soils. Starch grains can stay behind and be identifiable for thousands of years in a soil, along with all those other compounds. So we think of soils as being filled up with this brown, unidentifiable gunk. And that's actually largely not the case. A lot of the stuff in soil gets encrypted rapidly by microbial processes and encrusted with mineral particles. And then it could actually remain behind from DNA that we might be worried about because of genetic modification. We have no idea what that stuff does. It's probably fine though. To starch grains and all sorts of other things. So that's another interesting point about soils. Vitalists, exactly. Yep. So just to encourage you to get out there, one of my old grad school buddies actually managed to get a project together with Marcus, and they, well, Marcus, he's found it so much fun to be in the field with Marcus that he produced this movie, and so this really summarizes the thoughts that I'd like to leave you with at the end of this workshop, to go out there and just make it happen.
Well, that wasn't my youth. And if you all can see there were some of those coring equipment things that we were showing. That's the real take-home message. But no, as Troy said, you know, there's a lot of scope and a lot of fun to be had doing all of this too. In exotic places with a lot of food and a lot of good people. So, Chris, would you like to come and say some parting words?
Oh, I don't have any great summary to make, you know, and I'm not going to try to talk about which speakers were the best or which were the worst. Everybody was on time this year, so that was really outstanding. I really appreciate the efforts of the speakers who presented interesting lectures, who brought interesting material. Watching this from year to year, you guys don't know it, but I can see that this is not just the same old material rehashed every year. They modify and they bring in new information and new publications, and I can see how the talks have been changed every year, and I'm really grateful for the wonderful cooperation that we've had from each and every one of our presenters, mostly even getting their material in on time. And I want to thank each of you for coming along and for attending this workshop. I hope you got something worthwhile out of it. I hope you were, at some point during the last 2 days, inspired and maybe, re-energized to go back and do something more or different or change what you were doing before. And I want to thank Marcus and Karen, who is in and out, as the co-organizers. And so basically, I guess I just want to say, thank you for coming. Tell your colleagues that you thought it was worthwhile or not worthwhile. By all means, let us know what's worthwhile and not worthwhile. We really, 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 really want to have your feedback. So you may not have noticed, but there's guardians at the door. So until you hand your evaluation form in, you can't leave. So fill them out. Take your time. Do it right now.